Good afternoon and welcome back again, everyone. I am Birgül Çolak-Al and I'm a researcher at Bezmialem Vakıf University, Institute of Life Sciences and Biotechnology. I am working at Molecular Biology Lab with Dr. Metin Rafiki. Today, I will be host for this parallel session of CADD and SARS-CoV-2. I would like to announce housekeeping messages regarding our symposium. To ensure a smooth running of this session, please make sure you have access to a good Wi-Fi or cable internet connection. We kindly request that you change your Zoom profile name to your full name to ease name tracking for attendance purpose. The sessions will be live streamed on YouTube. For those who cannot get into the Zoom due to any technical issues, please join on the YouTube and inform us. Please join our Discord server for announcements and other communication. If you have any technical issues, kindly seek help through the chat box or email to us. If you would like to ask any question to our speakers, please type them in the chat. Now, I would like to welcome our chairperson for this session, Professor Dr. Atilla Akdemir, and our two honorable judges, Dr. Onur Serçinoğlu and Dr. Ayesha Fatima. I would like to confirm whether all the judges have got all their forms to judge. At the end of every presentation, there will be a 2-minute Q&A session. Now, I hand over the platform to Professor Dr. Atilla Akdemir. Over to you, Professor Doctor.
Thank you very much for the introduction. So let me give you a short info on how we're going to set up this meeting. In total, we have 5 oral presentations and 8 poster presentations. So for the oral presentations, you will have 10 minutes of time and 2 minutes of questions and answers. For the posters, it's 5 + 2. So let's start. I wish all the participants good luck with the presentations and with the QA sessions. So the first one, the title of this first oral presentation, which will be presented by Anuj Mavlankar, is titled Interaction of Surface Glycoprotein of SARS-CoV-2 Variants of Concern with Potential Drug Candidates, a Molecular Docking Study. So let's start the video. So, are there any problems? You good?
No, we are just opening.
Okay, now. I see. Thank you. There is no sound.
Okay.
Greetings to all. I am Anuj Mavlankar from Microbial Pathogenesis and Genomics Laboratory, ICMR, National Institute of Research in Tribal Health, Jabalpur, MP. My project topic is Interaction of Surface Glycoprotein of SARS-CoV-2 Variants of Concern with Potential Drug Candidates, a Molecular Docking Study. This is a graphical abstract of my overall work. In this work, I have selected the SARS-CoV-2 genome, that is the reference genome from Wuhan, and only the spike protein has been considered under study because it has shown some of the relevant mutations. So the spike protein has been randomly mutated with the amino acid or the amino acid positions that has been found to be mutated in all the variant of concerns that has been considered under this study, that is alpha, beta, gamma, delta and delta plus. in this study, basically, the variant of concerns has been considered because they are of utmost importance and has been declared by World Health Organization to be of important international concern. So once the individual mutations has been induced in the spike protein, the particular sequence of that variant has been modeled, structurally modeled using a Swiss model server. And following that, the model quality validation has been performed using certain softwares and tools like ProCheck, IRET, Save server of the University of California, and etc. among others. Next, the protein ligand has been prepared using Autodoc Vina tool for molecular docking study, and finally the molecular docking study has been performed using 5 drugs under consideration like baricitinib, favipiravir, lopinavir, remdesivir, and dexamethasone. So the basic objective of this study is to focus on the utilizing molecular docking to determine the effect of the selected drug targets on various variant of concern. So basically, in order to estimate the relative changes in the binding affinity of various potential drugs against the characteristic mutational profile of the spike protein sequence in different variant of concerns to predict the therapeutic efficacy of these drugs against the variant of concerns. So as we all know, COVID-19 has emerged as a global havoc. The virus, since its emergence from Wuhan in December 2019, the virus has undergone several structural changes due to mutations in its surface glycoprotein, also known as the spike protein. So these mutations have led to the emergence of different genetic variants or which poses great threat to the due to increase in their transmission and virulence, and they have taken several lives of innocent people all across the globe. So with this information, WHO has classified some of the genetic variants into 3 categories, which is variant of interest, variant of concern, and variant of high consequence. I have selected variant of concern for this study because they are at prevalence in the globe so far, and they have claimed so many lives. So just to check on what are the mutational profiles they have been, and what are the basic changes and changes in the structure and the functions of that strain that the virus has been going through, and what are the impact of the existing drug regime on these variant of concerns. So just to check it out, I have selected these drugs and the variant of concern for this study. So to ascertain the effect of the signature genomic variants like the single nucleotide polymorphism, as I stated, the insertions, deletions, mutation, so molecular docking is initial point to start with, and it serves as a good purpose to deal with this situation. So new drug development, as we know, is a long process. So repurposing of the existing drugs for their potential against the SARS-CoV-2 might be the current possible solution to mitigate the situation. This table represents the basic spike mutations that have been reported among different variants of concern. As you can see, D614G, it is one of the mutation which is prevalent among different strains of coronavirus. So this is the this might be the one which is consistent among different strains that has been evolving over a period of time. So as we know, surface glycoprotein is the one which gets attached to the human ACE2 enzyme present on the host receptor, and they basically promotes the viral entry into the host cell. It is divided into 2 parts like S1 and S2. S1 subunit consists of the receptor binding domain, which binds directly to the H, that is human ACE2 enzyme, and facilitates the viral entry into the host cell. This is the workflow I have gone through, like spike protein sequence, then followed by the 3D structure prediction, then the model quality validation, then the protein ligand has been prepared using Autodoc Vina, ADT MGL tools, version 1.2, then further the grid box has been prepared, and finally the molecular docking has been performed. So the top the topmost poses out of the total 8 dock poses has been considered for the study, followed by the molecular dynamic simulation. So these are the molecular docking results which I have got. In this, basically, the docking with the wild type, that is the Wuhan reference sequence, has been considered to be the 100%, and based upon that, the other docking postures between the drug and the variant of concern has been considered, and it's compared with the same. So as you can see, in the case of favipiravir drug, SARS-CoV-2 B.1.1.7, that is alpha variant, has been shown to be the highest efficacy, like 114.28% affinity has been shown. So it is of great significance. These are the basic best docking poses which has been shown with the drugs as well as the variant of concern. Next, the molecular dynamic simulation has been performed using the crystal structure and the equilibrated one. As you can see in the picture, the RMSD value between the equilibrated and the crystal structure is showing no much significant deviation between the two. These are the graphs depicting various procedures that has been incorporated while performing the simulation part using GROMACS, like the energy minimization, temperature equilibration, pressure equilibration, and the radius of gyration computation. So these are well in accordance with till 5 nanoseconds time period, but it has to be further made run so that the actual condition can be recorded. These are the superimposing images of the initial docking posture and the final docking posture of the spike protein. As you can see, the blue one is the initial one at 0 nanoseconds time frame, and the red one is the image at 5 nanoseconds time. So the RMSD value came out to be of no significance. That is why I have selected to ensure the run for further 10 or 20 nanoseconds so that actual picture can be reached upon. So the final conclusion of the study is that drug repurposing may help to discover and identify the existing drugs against the potential genomic targets of the novel coronavirus. So the drug candidates other than favipiravir and dexamethasone, they showed no significant alteration in the surface glycoprotein structure binding when compared to wild type, that is Wuhan reference sequence, implying that the current regimen of the approved drugs can be continued in the patients infected with these strains of novel coronavirus. So apart from that, molecular docking approach offers a great promise for predicting and shortlisting the potential drug candidates and is existing drugs for quickly reevaluating their anti-SARS-CoV-2 potential, which can help in the timely, effective interventions. Thank you so much.
Sorry, I was trying to get online. Thank you for the presentation. We have two judges, Aisha and Onur. So do we have any questions? Aisha, can I get the first questions from you if you have any? Aisha?
Yes. Okay.
Sorry, I have some internet problems also. So
sure. I'm Mr. Anuj. Are you there? Could you open your camera for a while? We can see you. Okay. Okay. Thank you so much. It was a very good, relevant study, I would say.
Thank you so much.
Okay. Could you move to your methodology slide, if we could see that again? If you can we go ahead? Oh, can you share your screen? Let me.
Yeah, sure.
Yeah, please share your screen.
Yes, sir.
Okay.
Is it visible now?
It's coming on. Okay. Could we move directly to your methodology workflow? Could you go to slide number 5?
Yeah,
Number
yes,
5,
ma'am.
6. Okay. Okay. All right. Then your molecular dynamics simulation was done on.
Gromax, ma'am.
Sorry if I didn't follow because we're doing something.
Ma'am, it's Gromax software. Is it audible? Am I audible to you?
Now it's okay. Now it's okay.
Ma'am, it's performed on Gromax.
Okay. And the force field was?
Force field is amber.
Oh, amber force field. Why amber?
Amber because I have read certain force fields for the simulation perspective, but I found to be more suitable for the purpose of binding. It consists of all the suitable parameters and the energy constraints therein. So I moved on to amber.
Okay. Okay. Thank you so much. I will give it to the second judge if he wants to ask the question. Thank you.
Okay, sorry, I am having some problems over here. So is Honor over there? Honor, do you have any questions? Honor is our second judge.
Anuj, thank you for the good presentation. I have one question also on your methodology.
Yes.
Can you go to the results where you showed binding affinities after molecular docking? Yes, so here you show, so here you show kilocalories per mole.
Yes, sir.
So they are all negative. So this is good. But then what does affinity percentage here means? So can you explain that to us?
Yeah, sure, sir. Basically, after performing the docking for the respective spike protein with the particular drug, basically the Wuhan sequence, that is the reference one, here it is shown as SARS-CoV-2 W3, I have considered as to be the 100% docking, though it may be for certain reason coming out to be low for some drugs as well. But I have considered for just to set a base benchmark for the relative comparison between different drugs and the variant of concern. I have considered the wild type to be the base one, and then with respect to that, I have compared it with respect to delta plus and alpha and beta, and for other variant of concern. So just to give a baseline value or of the docking.
Okay, thanks.
Okay, thank you very much. Unfortunately, I'm not able to ask any questions because we have a huge lack of time. We have already some delay due to problems. So I thank you very much for your oral presentation. Let's continue to our second speaker today. It is a poster presentation by Selvaraj, and the title of his presentation is Graph Theoretical Network Analysis and Molecular Modeling of 3,4-dihydropyrimidine Derivatives for enhanced cancer activity. So let's see the presentation, the poster presentation then.
Good morning. My name is Kunal Datta and I am a PhD student at Vidyasagar University, India. It is an in silico study of peptide analogs of the spike glycoprotein which showed antiviral properties against SARS-CoV-2. Last year on March 11, WHO had declared COVID-19 as a global pandemic. We are still in the middle of the battle against the SARS-CoV-2 with recent emergence of Omicron variant of SARS-CoV-2, which is now designated as variant of concern. An antiviral peptide is one of such alternative therapeutic strategy which are successful to combat SARS-CoV and MERS-CoV in previous studies. Therefore, with a similar kind of anticipation for the SARS-CoV-2, we have found 3 new antiviral peptides against SARS-CoV-2. Initially, the peptide sequences were predicted as antiviral peptide using ABP PRED and meta-ABP. Sequence 12, sequence 12M, and sequence 13M can bind the receptor binding domain of the SARS-CoV-2, which is a prerequisite for communicating with the angiotensin converting enzyme 2. The amino acid sequences of the predicted antiviral peptides were then mapped within the spike glycoprotein of the SARS-CoV-2, and the results showed that they are in fact analogous peptide of the spike protein. The molecular interaction between the ABPs and the viral membrane protein were likewise positive, indicating that it might hinder the viral repackaging processes. Recent work by Caron et al. Suggests peptide mimic of the human angiotensin converting enzyme 2 can block SARS-CoV-2 pulmonary cell infection with the high affinity antiviral peptides, namely P7, P8, P9, and P10. In addition, we have found that the predicted IC50 values of those high affinity antiviral peptides are about 41 to 42 micromolar. However, anti-SARS-CoV peptides, that is sequence 12, sequence 12M, and sequence 13M, showed lower IC50 value of about 38.54 to 39.38 micromolar, suggesting these ABPs, antiviral peptides, might be promising too. Results from the molecular docking studies shows that antiviral peptide and RBD had a thermodynamically favorable interactions. Moreover, after the insertion of
Presentation has frozen.
and sequence 12M revealed very low cytotoxicity. In addition, the molecular dynamic studies of ABP-RBD complex
Mohammed, can you assist? It has frozen again.
is
Hello, Dr. Attila. I'm sorry, I was told to stop because you were talking.
Oh, okay. No. I couldn't hear anything. That's right.
All right.
of 100 NS revealed a stable RMSD and RMSF throughout the MD trajectory, and they were most stable in case of sequence 12 and sequence 12M. In addition, The MMP binding free energy calculated of the different APP-RPD complex system calculated using MMPP or GBSA method showed very promising result in case of sequence 12M followed by sequence 13M and sequence 12M. In conclusion, the antiviral peptide sequence 12, sequence 12M, and sequence 13M could be helpful to fight against SARS-CoV-2. However, in future studies, these antiviral peptide could also help to develop anti-SARS-CoV nasal spray. Thank you.
So thank you very much for this presentation. This was Kunal Dutta. I was expecting this the second slide. This was the third presentation today. So I have some questions, if I may ask Kunal. Maybe I have missed it during your poster because there was a lot of how to say interference at this site. But can you discuss a little bit more detail the deep learning or the machine learning that you have applied, how you have applied it?
Professor, I think he's not here.
Okay,
Hello?
then continue. Is Kunal, are you there?
Yeah, actually, we did not build any simulation or machine learning algorithm. We actually use the previously published work or previously available the online server, that is Meta AVP and AVP Pred, that is based on machine learning. We did not program or code anything for machine learning, but we just use the previously available resources for prediction of antiviral peptides.
Okay, thank you very much. Aisha or Onur, do you have any questions? Otherwise, we should.
No, I'm okay.
Onur?
I'm okay.
Thank
I'm
you
okay,
very much.
Professor.
Okay, okay, thank you.
I do
very
not
much.
have any questions either.
Okay,
Thanks.
thank you very much. Mohammed, can we go back to the first poster, this graph theoretical network of Selvaraj? I announced him by accident, maybe.
Yep,
Is
sure.
that, okay.
Right.
Okay, so this is the poster by Selvaraj graph theoretical network. Yes, thank you.
I am Vajrala Achireddy, pursuing my third year in BTech Biotechnology from Kalasalingam Academy of Research and Education, that is in Krishnankovil, Tamil Nadu, India. And my co-members are M. Harish and R. Deepak. We are working under our professor, Dr. Selvaraj Kunijappan. And our topic of research is graph theoretical network analysis and in silico molecular modeling of bioactive compounds from Amaranthus spinosus against hepatocellular carcinoma. And our introduction: liver cancer is among the least successful treated of all cancers, but among the most readily preventable. Hepatocellular carcinoma is among the 10th most commonly diagnosed cancers and the 5th leading cause of cancer death. Currently available treatments for hepatocellular carcinoma is curative resection, liver transplantation, chemoembolization, and radioembolization, which have some drawbacks like treatment depends upon the tumor stage, patient performance status, and the liver function reserve, and requires a multidisciplinary approach so as to rectify all the drawbacks and to innovate a new idea on this area of research. And our main purpose is to provide a cure to hepatocellular carcinoma by non-invasive means, to provide affordable cost and efficient way of treatment in the field of oncology, to provide light on effective bioactive components present in our pioneer plant species of Amaranthus spinosus. And our hypothesis is we perform pharmacoinformatics based screening of bioactive compounds from Amaranthus species against the specific target HRAS. Amaranthus spinosus. We have identified 9 bioactive compounds through impact database, and they are the compounds are nicotinate, campestrol, cholesterol, heterothioconate, alpha spinastrol, beta carotene, 2,6 diaminohexaonic acid, phytosterol, and ascorbic acid. And we use different methodology in finding the drug. And they are: the graph theoretical network analysis was performed and identified ideal targets, that is HRAS, for in silico molecular docking by molecular docking using autodock VINA in Pyrex tool to predict high binding affinity bioactive compound. And the next is in silico analysis predicted ADMET and physiochemical properties of explored bioactive compounds and were used to access drug likeliness. Highly effective compounds were chosen by analyzing drug likeliness property and molecular dynamic stimulation. The first is graph theoretical and network analysis. The graph theoretical concept applied in multifaceted signaling network. It may be possible to systematically examine the topology and the functions of selected network. Further, the graph theoretical network analysis can be used to predict the structural and the dynamic properties of a selected signaling pathway. Such predictions can assist the selection of ideal drug targets for reducing the complication of cancer. It also gives data on the active sites and the molecular interactions of bioactive compounds, which may help in molecular docking study. And the main function of this RAS pathway, it is used to transduce signals from the extracellular to cell nucleus, where specific genes are activated for cell growth, division, and differentiation. This RAS pathway also involved in cell regulation, wound healing, and tissue repair, and integrin signaling, and also cell migration. The RAS pathway is able to stimulate angiogenesis through changes in expression of genes directly involved in the formation of new blood cells. Thus, signaling through the RAS regulate a variety of cellular functions that are important for tumorigenesis. Through the network analysis of this RAS signaling pathway, which we performed using CADD database, we found 3 nodes and 97 edges. And the next one is binding affinity of the compounds using autodock VINA. Here are the 9 compounds which we took. And of those, campestrol, it's showing high binding affinity, that is -9.1. Here we can find in the 2D diagram that campestrol is associated with many amino acids. In silico predicted ADME parameters and physiochemical of campestrol. Here, through this radar plot of campestrol, we can see the lipophilicity of campestrol is high. So we can see the insolubility also is high. If the lipophilicity of this compound decreases, insolubility also decreases. And it is the only lipophilicity that is violating the Lipinski rule of 5. Boiled egg model validation of campestrol. The egg boiled model is helpful to predict 2 key pharmacokinetic properties simultaneously. That is, the passive gastrointestinal absorption and the blood barrier penetration. The egg-shaped organization plot shows that the compound present in the yolk represent highly probable permeation, whereas the albumin represent highly probable human intestinal absorption. The compound and the standard egg found in albumin elucidate the good absorption of intestinal region. The toxicity studies of campestrol. AMSC toxicity and the max tolerable dose, HREG inhibition, LD, hepatotoxicity, carcinogenicity, skin sensitization, toxicity, and minotoxicity, all are listed here. In the molecular dynamic stimulation, RMSD graph, and the hydrogen bonding here are given here. The molecular dynamic stimulation study confirms the stability of HRAS ligand complex. The intermolecular interaction assumed by dynamic conditions paved the way to discover the bioactive compound campestrol and that prevent the cancer cell proliferation. RMSD is an important parameter that is used to analyze the equilibration of embryo trajectories and check the stability of complex systems during the stimulation process. RMSD of the protein backbone atom were plotted against the time to access its variation in structural conformation. And the hydrogen bonding, it shows how the bonding of campestrol with various amino acids. Here are the key findings. Out of the 9 compounds, the compound with higher binding affinity was found to be campestrol, that is minus 9.1 kilocalories per mole, and the same has been taken under consideration for validation. For validation, we plotted the compound characteristics in boiled egg model present in cis-ADME. And the plot, it was clear that the bioactive biocompounds taken with high binding affinity as a lipid compound. As a result, the lipid generally has a long-term effect while consuming it, and the same is needed for curing the cancer. And our conclusion is the molecular docking investigation exposes that bioactive compounds campestrol showed good binding energy against HRAS protein. The selected compound had no toxicity. The molecular dynamic stimulation study confirms the stability of HRAS complex. The intermolecular integration assessed by the dynamic conditions paved the way to discover bioactive compound campestrol that prevents cancer cell proliferation. Thank you.
Thank you very much for this oral presentation. Now let's quickly go to the question and answer session. So let's start with Aisha. Aisha, can I have some questions from you? Do you have any questions?
I'll give it a pass. I'll give it to
Okay.
you to ask.
Okay. Onur?
Hello, sir.
Yes, I have one question.
Yes,
Yes, sir.
please.
About your graph theoretical network analysis. Have you completed any kind of this target? Yes.
Sir, can't get you, sir. Can you repeat the question? Repeat that one.
If nobody has a question, I have a question.
Reptile network analysis. Reptile network analysis. Sir, can you repeat the question?
Should I repeat the question?
Yes, sir. Can you repeat it, sir?
About graph theoretical analysis. Have you completed any?
Sir, your voice is not correct, sir. Can you text it, sir? We'll answer for it, sir. Sure.
Okay, I text my question and you can answer in the chat.
Yes.
Okay.
So we are now waiting for the question of Onur, and after we get the answer of Vajrala, we will take a short 5-minute break. Have you seen the question, Vajrala?
Yes, sir. Yes, sir. Computer network matrix.
So have you computed any network metrics when performing your graph theoretical analysis? That's the question of Onur.
Sorry, KEGG pathway, sir. We use KEGG pathway and we track the HRAS protein, sir.
All right, thank you.
I think there is an issue with the connection, so maybe we should continue to our break, and the question can be answered by texting back. Shall we do it like this, Vajrala? If you can write Onur back with your answer, it would be great. Yeah. So let's take a 5 to 10 minutes break. We will update you on the progress and then be back here again. So Virgül, the floor is yours.
Okay, thank you, Professor Dr. Attila. We are now taking a 5 minutes break. See you in 5 minutes. Hi everyone, welcome back again. Now we will continue our poster presentation. Now I hand over the platform to Dr. Attila. Please.
Okay, thank you very much. Welcome back, everyone. Sorry for the very short break that we had to schedule in. We had some small technical issues that we fixed, I assume. So we will continue with the poster presentation of ESMA. And the title of the poster presentation is In Silico Analysis of High-Risk Missense Variants in Human ACE2 Gene and Susceptibility to SARS-CoV-2 Infection, as you can see on our screen. So, well, the floor is yours.
Hello everyone, I would like first of all to thank the organizer of this meeting for inviting me here today. So let me introduce myself. I'm Seh Asma and I'm a third year PhD student from Hassan II University in Casablanca, Morocco. So I'm pleased to present my poster on in silico analysis of high-risk missense variants in human ICE2 gene and susceptibility to SARS-CoV-2 infection. So I divide my poster presentation into four main parts. So the SARS-CoV-2 colonizes the respiratory tract system, causing symptoms similar to those of common cold, such as respiratory disorders, runny nose, cough, shortness of breath, and fever for several days. So SARS-CoV-2 infects the respiratory tracts and engender acute respiratory disease, and generally it resides in angiotensin-converting enzyme 2 as an entry receptor for SARS-CoV-2 spike protein priming. So, very recently, it remained possible that ICE2 gene polymorphism may influence both the susceptibility to SARS-CoV-2 infection and COVID-19 disease outcome. So, the purpose of this presentation is to show you the deleterious variants in ICE2 gene by using various bioinformatics tools to extrapolate the possible associations of ICE2 polymorphism with COVID-19 susceptibility. Then I will move on to the materials. In our study, 242 missense variants were collected from ensemble database. After that, SIFT, Polyfen, Proven, and Panther algorithms were used to predict the functional effects of mutation on the protein. Secondly, MoPro and iMutant tools were used to identify the mutation effect on protein stability. Thirdly, Conserve, Swiss model, and string were used to predict the mutation effects on protein structure, function, and protein-protein interactions. So let's now move on to the results. So in our in silico analysis, we identified 6 non-synonymous SMP, namely D427Y, R514G, R708W, R710C, R716C, and R768W from 12 non-synonymous SMP. The 6 tested non-synonymous SMP were predicted to be deleterious by at least 6 algorithms, decreasing the stability of ICE2 by both MoPro and iMutant, and located in conserved regions with a score conservation of 9. Expected D427Y, which may affect the structure and function of ICE2 protein. Then the interaction analysis using string server suggests that the ICE2 protein not only participates in hypertension and cardiovascular disease, but candidates also in COVID-19 disease. So lastly, these findings show that the 6 tested variants are the structurally and functionally most significant variants in human ICE2 gene. So it has been found that the residue arginine 708, 710, and 716 are located in the dimeric interface of human ICE2, and they are found to be important for its cleavage by the protease TMPRSS2, and this processing is indispensable for augmentation of SARS-CoV-S driving entry into host cells. So finally, I would like to address the limitation of our in silico analysis is that the 6 deleterious variants should be confirmed with future extensive experiments and clinical wet lab approaches to figure out the mechanism of these mutations in susceptibility to COVID-19. And thank you for your attention.
Thank you very much, Esma, for this presentation. Would like to have some questions. Are you there? First of all, if you're there, can you show me figure 3, please?
You can ask Dr. Tila.
Okay, yeah, I wanted to see figure 3, whether Esma can show me figure 3. I have a question about figure 3. Esma, are you there? Okay, she seems to be not here. So for the sake of time, we have to continue then to the next poster.
Dr. Tila, I think she put in the chat, she's here.
Okay. But I don't hear her. Okay, she has a connection problem. So we need to continue. And if she can restore connection, we can ask the questions to her. Is that okay? So let's continue with the next poster. So please write me, Aisha, Esma, if you are here, if you can join us, if you restore your connection, please write me and we can schedule a new the question hour. So now let's continue with the next poster. In silico and in vitro elucidation of Balactaka compounds on macrophage polarization. Yes, it's on the screen. So yeah, we can start the video now.
Hello everyone, I am Vijayalakshmi Muniyandi from India, presenting the poster on the in silico and in vitro elucidation of Balactaka compounds on macrophage polarization. Initially, inflammations are otherwise known as boosting the immune system. Macrophage polarizations were studied in this research, which is nothing but an alteration of the expression of the macrophages based on their different phenotypes, M1 and M2 phenotypes, in which M1 is involved in the pro-inflammatory actions and M2 are involved in the anti-inflammatory actions. In this study, we have chosen a plant compound Balactaka for the macrophage polarization study. The research studies were carried out in the following steps, such as retrieving the bioactive compounds and then identifying the target proteins, followed by a molecular docking study, then admit property prediction, finally an in vitro analysis for the macrophage polarization. The result predictor here is the identification of the target protein through the graph theory by the cytoscape software in the TLR signaling pathway. The proteins identified were the NF-kappa B and the phosphoprotein, NF-kappa B and the phosphoproteins. Can either induce or inhibit the proteins in case of any inflammations. The proteins identified were modeled using the Swiss model server using the protein sequences of human. Then the bioactive compounds, 17 bioactive compounds were identified from the research reports, and together the bioactive compounds and the target proteins were taken to a docking study. The docking study were carried out using a Pirate software, and then they uses an autodock VNOT tool to dock the proteins with the compounds. The compounds such as L+ascorbic acid dihexadecanoate methyl tetradecanoic acid has the highest binding affinity -4.7 kilocalorie per mole with the NF-kappa B proteins, and then another compound hexadecanoic acid has the binding affinity of -3.8 kilocalorie per mole with the phosphoproteins. The result predicted from the molecular docking study indicates that the Balactaka compounds has the greater binding affinity to the NF-kappa B protein than the phosphoproteins. Then an admit property was studied. ADME property was studied using an Swiss ADME server, and then toxicity study were carried out using a PKCSM software. Then finally an in vitro analysis were carried out for their macrophage cell viability and their morphology. Finally, polarization studies were carried out. The cell's viability were assessed using an MTT assay in which the differential concentration of the Balataka crude extracts were treated to the cells, and the cells were identified at a different absorbance at a 595 or 655 nanometer. The results depicts that the Balataka extracts of 45 microgram per mole has the highest absorbance than other concentrations. The visualization of the macrophage through the phase contrast microscope indicates that the treated cells changes in their morphology due to the cytoplasmic elongation that causes the macrophage polarization or their change in their phenotypes. Then finally, a macrophage polarization studies were carried out in the cell line. The cell lines were treated with control and then Balataka crude extracts. This macrophage polarization studies were carried out at an interval of 8 hours. The picture shows here that there is a migrations of the immune cells towards the tissue that is made to wound. And at the 24th hour, there is a massive migration of the macrophage cells at the site of wounded area. In the control tissue, there is also a migration of macrophages, but the time taken to attain to heal the process is very less when compared to the treated cells. Thus, from the summary of the poster, I am presenting that the Balataka compounds has the greater binding affinity to the NF-kappa B protein as studied by the molecular docking study. This NF-kappa B proteins will initiate the secretion of the pro-inflammatory cytokines. The admin property studies indicate that these Balataka compounds can be used as a drug. Further studies on the in vitro also suggest that these Balataka compounds induces the macrophage polarization, which activates the immune cells through the NF-kappa B. Therefore, the healing process for the wounded area is very less when compared to the normal process of immune cells. Thus, these compounds can be used in case of the immune suppressing host to induce the immune cells and to heal faster than the normal persons. These are some of the references for this study. Thank you everyone.
Thank you very much for this poster presentation. I have one question, if you're here. So you performed the docking against two targets, NF-kappa B and the phosphoprotein, and then you share some binding, strengths binding, affinities binding, energies, I should say. Are you confident that this docking, the scores that you just shared are correct? Why are you so confident on these docking scores? Can you share it with us, please? Are you here?
Yes, sir, I'm here.
Okay.
It's just an in silico study, sir. Though these compounds, these proteins are identified from the graph theory, so they may have the binding affinity to this particular proteins, the compounds from the Balataka.
Okay, but you then go to docking studies to investigate the binding, basically to understand the strength of binding. And do you believe that docking studies are sufficient for this? Are they reliable enough?
Yes,
especially for if you look at 17 compounds?
Yes, sir. For that further, we have carried out the in vitro studies. So from this in vitro studies, we confirm that NF-kappa B may be the target proteins.
Okay, thank you very much.
Yes.
Any other questions, Aisha, Onur?
Yes, I have one.
Yes, sir.
It is highly related to this previous question, actually. So do you believe that your in vitro results could have resulted from binding to other proteins?
Binding, but the other proteins have these proteins have the higher degree of targeting the genes, targeting the transcriptional factors. So out of the 17 compounds, these compounds have the higher binding affinity. So these compounds may have the effect in the cell lines.
Okay, thank you.
Okay, thank you, sir.
Thank you. Aisha, do you have any?
It's okay. You can.
Thank you very much.
Continue.
So let's continue for the sake of time. Our next presenter will be Eda. She will have a oral presentation entitled Benchmarking Why to Use Structure-Based Binding Energy Predictors on Spike RBD ACE2 Deep Mutational Scanning Dataset. So if we can, here it is. Okay, let's play it then.
Hello everyone, I'm Eda Şamiloğlu. I'm a master student at İzmir Biomedicine and Genome Institute and our lab located at İzmir Biomedicine and Genome Center. First of all, I would like to thank the organization committee of the symposium for their effort and provide us the opportunity to present our work. Today I will talk about our benchmarking study of structure-based binding energy predictor on SARS-CoV-2 spike and human ACE2 deep mutational scanning dataset. First, I will start to talk about SARS-CoV-2 variants. Scientists and even not scientists watch SARS-CoV-2 variants with eagle eye since circulating variants emerged new situations like enhanced transmission. And then we will take a glance at experimental effort, which name is deep mutational scanning, to reveal mutations effect on SARS-CoV-2 spike and human ACE2 binding. Then we will focus on to the in silico approaches, those we use in study. And lastly, I will present benchmarking results, our findings and conclusion. Here you see the receptor binding domain, shortly RBD of SARS-CoV-2 spike with orange and ACE2 protein with blue, which initiates the viral infection. At the right hand side, you see the interface of the protein complex where binding happens. Not surprisingly, SARS-CoV-2 variants has various mutations in the interface. The first variant alpha has a mutation at the 501 position, convert from asparagine to tyrosine. This mutation is very popular because it's first reported interface mutation. Then beta, gamma, eta, iota, lambda, mu, and lastly the Omicron variant were reported. Omicron draw attention of scientists since it has 9 interface mutations. The interface mutations of SARS-CoV-2 variants poses a significant importance in terms of binding affinity and transmissibility change of virus. To this end, there were a huge effort for calculating binding energies of SARS-CoV-2 variants. Here we move to the experimental efforts to reveal mutations effect on binding. By using the mutational scanning, ACE2 and RBD interface was scanned by point mutations and binding affinity change upon mutations were reported. Blom et al. Scanned RBD interface by converting the wild-type amino acid to the 19 possible amino acids for nearly 200 positions. And for ACE2, Proco et al. Presented same kind of dataset for ACE2. This dataset maintains importance during the pandemic for variant effect and therapeutic studies. However, deep mutational scanning is time-consuming and costly method. For example, the most popular mutation at 501 position, wild-type amino acid and converted to the Y. After mutated RBD were produced and localized in the cell incubates with ACE2 and binding complex sorted by FACS. And these experiment sets repeats for each mutations, including the controls. For the calculating binding energy change upon mutation, simply wild-type binding energy subtract from the mutant energy, and according to that, negative delta G means enhanced binding affinity, and positive delta G means decreased binding affinity. Scientists revised to these deep mutational scanning dataset after each variant, especially after the Omicron. Some scientists look for the in silico approaches that can calculate delta G accurately and fastly. Some of them published their studies as a preprint and posted on Twitter. After that, another scientist critically commented to those tweets. Some ones mentioned deep mutational scanning studies of Blom et al. Some of them underlined solving mutation effect on function is not easy problem to solve by even experimental binding energy. Some of them mentioned docking cannot predict binding affinity reliable for most cases. So the question is that can in silico approaches predict how mutation affect binding affinity? For an in silico study, mutations model on 3D structure first, then the model score by using scoring function, then delta G calculates that I previously described. In our study, we benchmark 6 different structure-based delta G prediction approaches to answer the important question. We use HADOC and UEP as a naive predictor since HADOC has simple energetic scoring function and UEP scoring functions build on contact terms. Addition to those, we use FoldX, EvoF1, Mutabind2, SSIP predictors. FoldX scoring function contains physical energy terms and it has an option to calculate delta G with and without water. Also, EvoF1 has physical energy terms in the scoring function. And Mutabind2 and SSIP contains both physical energy terms and evolutionary conservation terms. We benchmarked those widely used binding affinity predictors on 263 interface mutations. We selected positions that only actively contribute to the binding. Here you see the distribution of our data set. We selected half of mutations from enriched cases and another half from depleted cases. Enriched terms used to specify enhanced binding affinity and depleted vice versa. Here you see the selected mutations for RBD and ACE2. Each spot color according to the experimental binding affinity value. Also, positions located in the y-axis and possible amino acids on the x-axis. For instance, N501Y, this spot represents results of a whole experiment that I explained before. We revealed performance of those predictors by comparing with experimental binding energies. FoldX has the highest performance on overall dataset and performance of FoldX and FoldX water are not different that much. So we can say that water contribution do not affect FoldX performances in this case. Also, overall performances do not differ significantly among the predictors. Addition to the overall performances, we present predictor performances on RBD and ACE2 separately. Again, there is no big differences. However, when we analyze performances on enriched and depleted cases separately, Mutabind and SSIP perform differently. Addition to those findings, we present predictors performances on highly packed residues, which are selected by UEP. For this analysis, we have 129 case in dataset. Interestingly, EvoF1 show very low performance on highly packed enriched cases. To explore predictors' nature deeply, we built metric analysis. Let me explain those metrics on an example. Here the example, mutations from N to Y. We classified mutations by using its van der Waals volume change. This matrix calculates similar to the delta G calculation. Volume change calculates by subtract van der Waals volume of wild type from mutants. Also, hydrophobicity and flexibility change calculates by using same way. We presented results of those metric analyses in this figure. For better present, we investigate this figure by using delta success rate. Delta success rate is difference of enriched and depleted success rate. Dark red and blue represent extreme delta success rates and tell us about biases of predictors. Mutabind2 and SSIP has notable delta success rates for each matrix. In fact, this originates from depletion bias of these predictors that we previously mentioned. Addition to that, we found that HADOC predicts most of the volume increase mutations as affinity enhancing, and for FoldX predicts mutations with decreased hydrophobicity as affinity enhancing. We built root median square deviation shortly RMST analysis for those models. RMST is the standard measure of structural distance between coordinates. We aligned our structure on the reference structure. Here the example is again N501Y. Let's look into the interface closely. N colored with gray and in here Y modeled by Foldix, you see magenta. Similar to that, SSIP model, same mutations in a different orientation. And here we see all modeled Y amino acids here. We reported these analyses for all mutations in the dataset, and we found that hedox structures differ from others. We think it's because of hedox refinement step. In this study, we found that Foldix and EVF1 have first and second highest performances respectively. Our performance analyses show that Mutabind2 and SSIP bias toward predicting most mutations as depleting, and Foldix predicts mutations with decreased hydrophobicity as affinity enhancing. Also, HEDOC predicts most of the volume increasing mutations as affinity enhancing, and models of HEDOC differ from others because of its refinement step. In conclusion, our system-based analysis show that classical affinity predictors are not yet fit to predict the binding affinity change of a virus host system like SARS-CoV-2 ACE2. My last word that I would like thanks to my PI, Dr. Ezkarac, and my colleagues who has wonderful contribution to this study, Melis, Burcu, and Mehdi, also all members of Karaca Lab, and of course to you for listening to me. For further news from Karaca Lab, you can follow us from Twitter and GitHub. Thank you.
Thank you very much, Eda, for this nice presentation. Well, let's go to the question section. Aisha, can I have your questions for Eda?
No questions, but yes, it was a very good study. Okay. I was thinking first I couldn't follow what was happening, then I guessed, then I followed, it was okay. So you were trying to compare the different programs, is it?
Yes, 6 different programs.
Just the comparison of different programs, what kind of predictors they would give, or they are prediction programs, but what is the effect of them, is it?
Their performances, we worked on their performances by using experimental delta G values. We already know the experimental delta G, so we can look for for performances of these tools, and then we see that the performances overall not change, but when we dissect the data set according to enriched depleted mutation or according to volume change, we see something that about nature of predictors.
Okay, thank you so much.
Okay.
Onur, do you have any questions for Eda?
Yes.
Please.
First, Eda, this was very good. Thank you. I have one question. If you go to your conclusion slide, can we do that?
I cannot share my screen, but maybe someone can do for us.
Just give me a second. I'll make you a co-host and you can do share. Eda, can you try, please?
Okay. Just a second. Can you see?
Yes. Oh, so that sentence. No, the next one, I think the next slide. Yes, so classical affinity predictors are not yet fit to predict binding affinity change. Can we conclude that none of them are successful enough for practical applications? What do you think?
Let's say Foldix has the highest performance, but it is not even fit because it has, for example, hydrophobicity bias. So it tells us there should be a bias while we try to predict. So let's say we see that MD analysis, some MD analysis for a delta G prediction are significantly have good performance. So I didn't mention these studies, but its affect our conclusion.
Okay, thank you. Thank you.
Okay, thank you, Eda, for your nice presentation. The next one will be a poster presentation again. In fact, we will have two succeeding poster presentations. The first one being presented by Pranavati Yana, target discovery and animation of hypothetical proteins from Klebsiella pneumonia, identifies BCT, family protein, and so on and so on. Yes, that's the one. So good luck. You may start.
Hello everyone, I am Pranav Dhyani, PhD student from Department of Bioinformatics, Pondicherry University. This is my poster presentation video entitled Target Discovery and Annotation of Hypothetical Proteins from Klebsiella pneumoniae, which identifies BCT family protein involvement in host pathogen interaction. Klebsiella pneumoniae is a gram-negative opportunistic bacterial pathogen that causes various healthcare associated infection in immunocompromised patients. This pathogen has become resistant to various currently available antibiotics, including some of the last resort treatment options like colistin and carbapenems. In 2017, WHO released a list of priority pathogens for which urgent antibiotics are required. And also recently, India released a priority pathogen list which includes Klebsiella pneumoniae as a top priority pathogen. To tackle the pressing issue of antimicrobial resistance, new interventions are required in discovering novel targets as well as new antibiotics. In this context, hypothetical proteins are proteins that are predicted or identified protein but lacks experimental evidence in functional role. About 20 to 40% of newly sequenced genome consist of hypothetical proteins, and which are difficult to characterize experimentally, and primarily they are annotated using computational approaches. This can aid or assist in identifying new functional role as well as identifying unexplored targets in an organism. To identify and annotate the hypothetical protein, we followed an overall methodology comprised of several steps. First one is identification of hypothetical proteins from the genome or the proteome actually, and then screening of proteins which has the properties of target likeness, like that is essential to the pathogen and non-homologous to the host proteome, that is human, and function prediction of those proteins was carried out by domain analysis, gene ontology analysis, and then interaction analysis. And we were able to characterize 17 of such proteins, and further physicochemical properties, virulence prediction, druggability, subcellular localization was carried out for the 17 proteins, and further host pathogen interaction of the 17 proteins was carried out, and one protein was identified to have interaction with human host, and for which a structure of the protein was predicted. So the overall finding are 540 hypothetical proteins were identified in the genome of Klebsiella pneumoniae, out of which 40 proteins were found to be essential, which are similar to E. coli, Vibrio cholerae, Salmonella, and Mycoplasma. And out of the 40 proteins, 30 proteins were non-homologous to host. That is, these proteins are exclusively present only in the pathogen. And we were able to annotate successfully 17 proteins, and all these 30 non-homologous proteins has the capability to become targets, and the functions of these 17 proteins ranges from like enzymes, lipoproteins, stress-induced protein, transporter, toxin proteins, etc. And the virulence prediction of these 17 proteins identify two proteins which are similar to Listeria monocytes protein P60, which is an extracellular virulence protein which helps in survival and host invasion, and the other protein is similar to E. coli protein, which is also an extracellular adhesive fiber common protein which is present in both commensal and pathogenic E. coli. And the 17 proteins were classified based on their localization into drug and vaccine targets, and 16 of the protein which are present in cytoplasm, periplasm, and inner membrane are classified as drug targets, and one protein as a vaccine candidate. And druggability analysis from targets present in drug bank and CHEMBL showed that these proteins are indeed novel targets, and further characterization of host pathogen analysis identified BCCT family transporter protein, which is similar to Escherichia pestis protein, having interaction with 5 of the human host protein. The human protein has the functions ranging from binding, catalytic activity, transcription regulation, and biological adhesion. The spectra of that protein which is involved in host pathogen interaction was predicted. So the conclusion of the study is understanding the function of hypothetical protein is important as it facilitates to identify new roles of proteins which are previously existing, and also aid in identifying new therapeutic targets. And 30 of these pathogen-specific essential proteins could be considered as novel targets, and 17 proteins were successfully annotated using computational approach. And all these targets are novel targets, and then 16 of them are drug and one is vaccine candidate, and that was involved in host pathogen interaction. And the structure prediction of these proteins was carried out, and these overall findings from the study could help in developing new antibiotics as well as new treatment measures in tackling Klebsiella pneumoniae infections. Thank you.
Thank you for this presentation. I have a quick question. So can you discuss how you define druggability, how you have implemented this into your project?
So in this project, we have used existing targets which are present in drug bank and CHEMBL. It is more like a sequence-based approach where we have not included the structure models, but based on the sequence similarity. So if they show high similarity, so we assume that there is a drug target present in the database. So we assume they are druggable targets. Otherwise, they are like novel targets. Mostly they are like FDA approved targets will be present in drug bank or investigational targets.
Okay, thank you very much. Aisha or Onur, do you have any questions?
Yes, I have one question. So you have used a sequence-based approach to determine similarity between known drug targets and your hypothetical proteins. Is this correct?
Yes, that's right.
So do you have any similarity level results, or did you have a cutoff for this similarity? How did you decide whether your protein was actually similar to known targets?
Okay, whatever we found so far in the study, based on the annotation, these 17 targets, right? So annotated proteins were not similar to the existing targets actually. We didn't find any similar target sequence present in either of these database.
All right, thanks.
Thank you.
Thanks. Aisha, if you don't
Do we have time? Do we have time? Then I have a question.
For you, yes.
We have a question. We can ask, right?
Yes.
Okay. I couldn't understand your methodology quite clearly. If like Dr. Attila and Dr. Onur said, how did you do it actually? Could we see your targets, or can I go see your target if possible? Do you have this on your computer?
Currently, I don't have. Maybe later I could show you or send you.
Yeah, sure. Yeah.
I can tell that these proteins were similar to known pathogenic proteins, like as I mentioned, E. coli and other species of bacterial pathogens.
Okay, thanks. Thank you, Attila. We can continue.
Okay, thank you very much. We continue with another poster presentation by Jamil, entitled Complete Alpha-2 Model Assessment of the Ryanodine Receptor Isoform 2. So floor is yours.
Hi, I am Cemil Can Saydan. I am a master's student in Computational Science and Engineering Department at Istanbul Technical University. We work on the computational biology studies, and in this study, we try to build a complete model structure for random receptor 2 using the state-of-art model IFOL2 and other methods of TR Rosetta and Rosetta comparator modeling. And random receptors are homotetramic intracellular ion channels, which are key element in excitation and contraction coupling by regulating the calcium release from the cytoplasmic reticulum to cytosol. And there are three isoforms, and we are working on the random 2, which is most abundant in cardiac muscles. And random receptors have a common mushroom-like shape, consist of a large cytoplasmic head and the transmembrane part. And aberrant random 2 activity causing the heart disease, such as a tachycardia or arrhythmia. And previously, several different random 2 structures have been released, and yet there are missing regions in the structures. And in our methodology, we applied the divide and conquer principle, which means that we focus on the missing region individually, then we combine all missing structures into the one final model. Before we start the modeling, we analyze the PDB sequence of the available structures, and we see that the most of the missing sequence amounts varies from the 40 to 100, except for this region actually. And this is the largest missing region, which is located near the transmembrane region. And although we tried to model this region, we failed, and in our results, and I didn't show up here, but we see that this region might be embedded in the membrane, or it's highly disordered. And also previously for random 1 modeling, the equivalent of this region was not modeled for full-time simulations. And in the result, the channel's function is still correlated with the experimental data. So for now, we did not include this region in our final model. And let me explain our approach for showing our results. For this region, which is here, first we applied the IFOL2, and it failed actually. And there might be 2 reasons for this. First one, we have a low amount of MSA data, multiple sequence alignment data, which is the 17 actually, which was the 17. The second one, this region is a disordered region actually. And so we applied the TR Rosetta protocol, and it seems a contact between these 2 regions in the structure. And after that, we applied the 500 nanosecond molecular dynamic simulation, and we see that this contact still exists at the end of the simulation. And after that, we keep this structure in our final model, and this is our final model. And for this region, previously we applied the comparative model. And after the IFOL2, unlike the crime structure and the random receptor, unlike the crime structure of the random receptors, IFOL2 puts this structure into it showed that this region as a part of the IFO7 motifs in here with very high accuracy, which actually causing the structural shift for this region actually. And for this region, we applied a similar approach. Before the IFO2, we applied the comparative modeling, and this region actually kind of complicated because there are several different regions coming together, and there are conflict in the crime structure, and we are not sure the structure and also the sequences. After we applied the IFOL2, we see that there are the structural shifting in the crime structure. And when we directly use the crime structures, we see that there's the shifting in here. And in the conclusion, we see that At the end of in conclusion, we combined all of this structure in our final model, which looks like here. I put our final model, and we see that we clearly saw that the IFO2 highly occurred in our protein compared to the other methods, and we are currently running a Phylogenetic Protein 2 simulation to optimize this structure. And this is our study. Thank you for listening.
Thank you very much for this presentation. Aisha, Onur, do you have any questions? Aisha?
I'm okay. Just a comment, it was a good one. However, I couldn't really get to the comparison of your structures that you were doing. You were showing on the cryo and then on the AlphaFold, correct? So somewhere I got lost somewhere.
Yeah, actually, I suppose I need to represent my study on my poster. I didn't prepare the nice presentation. That's why I couldn't show all information at exactly the cryo structure, because we have limited area for poster. But let me explain. Can you share my poster maybe? Yeah, for example, let me explain in the cryo structure, I some area, but the remaining of the structure directly I get from I restrained and I directly, it's similar to cryo structure. I mean, maybe, sorry for.
Are you sharing your screen?
Yeah, I'm looking now. Slides of mine for your sorry for
If we have trouble, we may continue.
yes, please. I couldn't my slides actually.
Okay. Aisha, any more comments? Otherwise, shall we go to Onur?
I think you can go on. No problem. We can find
Onur?
something later.
I don't have any. Thank you.
Okay. Thank you very much for this presentation, Onur. So I'm sorry, Jennifer. Thank you for your presentation. Now I'm continuing with our next candidate, Sefer, but I also from Istanbul Technical University. I see the oral presentation already online. So, well, the floor is yours, Sefer.
Hello, my name is Abdul Salam Yazid and my advisor is Sefer Badai from the Informatics Institute at Istanbul Technical University. I will talk about Swift docking, which is a neural network-based method for speeding up molecular docking. Traditionally, virtual screening is used in selecting the top molecules that will bind onto a target protein. It is used in narrowing down such space of millions of molecules, and it is crucial in drug discovery. It is divided into ligand-based and structure-based. In the case of structure-based, docking is performed. Nowadays, the size of data is increasing rapidly. For example, the zinc database contains 230 million molecules. And to process huge amount of molecules, you need a HPC environment, high performance computing environment. In this work, we aim to accelerate docking of large libraries using machine learning. There are works that rely on machine learning to predict docking scores. For example, lane docking, emulating docking and deep docking. In the case of lane docking, 10,000 molecules are selected out of millions and used to train a model which will be used to make predictions on a separate set of ligands. Emulating docking works similarly. Deep docking uses millions of molecules for training machine learning models, and it involves constant redocking in the training process. In our work, which is Swift Docking, uses a long short-term memory LSTM neural network, and it is designed without the need to be used without any HPC or any GPU. And all you need to provide is a CSV file containing your ligands, smiles, and the docking scores on a target protein. And in our case, for example, if you provide the ligands, say you have millions of them, in our case that we have up to 3 million molecules for a target, we select 0.2% or 7,000 molecules and train a neural network, and then use the network docking scores prediction on the 99.8%. Well, if you provide a CSV file that has smiles, those smiles, you just cannot push them into a neural network. You need to convert them into something numeric, and that's why we have a set of descriptors that we used in our experiments. So some of them are distinct. For example, one hot encoding. Some of them, for example, D3 contains Marquis fingerprint, which is a 167-bit vector that asks questions about a ligand. For example, if there is a carbon atom, it puts 1, else it puts 0 in a particular place in the long 167-bit vector. On the far right, we can see the number of features for each of the descriptors. We have 3 target proteins. Actually, the first one and the third one are the same target, only different binding sites. And that target protein is a DRP1 GTP base. In the first case, it is a GTP binding site, and the second case, it is M1D 49 interaction interface. The second target is a ryanodine receptor, and the binding site is a dandrolin binding site. On the far right, we can see the number of ligands in each of these cases, in the 3 targets. We use Autodoc Vina for the docking process. So after, during the training, we experimented with different combinations of descriptors with different training set size to see the effect of the training set to train the model. So out of, say, 1 million molecules of a target, we select 7,000 for training. We also select 10,000 up to 250,000 for different descriptor and for each of the targets. And we calculate the R-squared coefficient of determination on a test set. This R-squared metric is between 0 and 1, and R-squared of 1 means the model is perfect. The predictions of the model's docking scope predictions are perfect. 0 means the model is not generalizing well. So, for example, here we have for training on target 1 and 7,000 ligands out of 3 million, for example, the testing data R-squared is 0.83, and for 350 it is 0.86. The difference is not much, which made us conclude that it is okay to just use 7,000. So we also plotted the scatter plot of the 3 targets between the predictions of the model's predicted docking scores and the actual docking scores for different models for the 3 targets using the D3 descriptors and training on 7,000 molecules. And the far left, we can see the Swift docking LSTM. Other models are decision tree, stochastic gradient, Treason regressor, and XGBoost. Visually, the long short-term memory perform the best. The perfect prediction and actual docking scores scatter plot will be a straight line. So and this is for all of for target 1. The first row is for target 1, second row is target 2, third row is target 3. Decision tree in general performed the worst, and the target 3 was for all the 3 models, even for our LSTM model, was the one performing least in terms of it was also performing the worst. So finally, we believe this system can significantly improve virtual screening VS, and our finding is that our realization is that the descriptors are very important, and we have designed the system with ease of use in mind. You don't need any GPU, you don't need any HPC environment, and we will also provide the code on GitHub so that everyone can have access to and try. All you need to provide is a CSV file with the smiles and their docking scores. Thank you very much.
Salam, thank you very much for your presentation. I may have a question if you're here. Okay.
Yes, I am.
Okay, so can you comment on the descriptors? It's of course a very complex set containing hundreds and thousands of descriptors, of course. But so can you try to comment on what kind of descriptors they are? What's the difference between D1, D2, D3? So we can try to imagine what is this all about. Thanks.
Okay, so first of all, if you have a smile, like for a ligand, you just cannot use the smile on, say, a neural network. You have to convert it into something numeric. So we experimented with different combinations of different fingerprint of a ligand smile. So not only that we tried different combinations, we tried concatenating different fingerprints to make it just one fingerprint. So this, in the presentation, we have D1, D2, D3, and D4. Some of for example, D3 is a concatenation of different fingerprints. And in the concatenation, we have Marquis fingerprint, Morgan fingerprint, one-hot encoding, for example. So this was actually what we spent huge amount of time on, experimenting with different combinations to find the best results.
Okay, thank you. Maybe one more question.
Okay.
These descriptors are all ligand-based, of course, but is there also a way, or can you comment on possible ways to also include information of the binding site or ligand-protein interactions in these descriptors? Are such descriptors known? Is it possible? Have it been tried? Do you know this?
No, okay, so we did not try that regarding the binding sites. I mean, any descriptor about binding sites. We did not perform that in our experiments.
Are you aware whether they are available or whether they have been performed by others?
Well, there is the library that we use. It's called DeepChem, and it has so many, I think up to say 30 to 40 different type of descriptors. So it was hard for us to, of course, experiment with all of them with different combinations, but there are certainly some descriptors that we did not perform experiment on, maybe related to your question about maybe the binding sites.
Okay, thank you very much.
Okay.
Thank you very much. Aisha, Do you have any questions?
Just one question. How did you collect the actual docking scores? Predicted, I can understand. Where did the actual come from? Actual
we use a program, AutoDocVina.
But that's a prediction.
No,
How did you?
Prediction is our prediction from the machine learning model. And the
no, what I want to know is that your one of your slides before the conclusion, the last slide said predicted scores versus actual scores.
Yes.
So
so when we say
where did the actual scores come?
When we say the actual scores are the ones from the AutoDocVina program. And when we say predicted, we mean the ones predicted by our models.
But then AutoDocVina also predicts. Then what's the point? Why? I can't.
So you see, docking is in AutoDocVina, you have so many ligands and you have a target, and you have to find the docking score of, say, millions, maybe 1 billion ligands on a particular target. So instead of doing that, we pick just maybe 7,000 of, say, 5 million, and we train a model using that 7,000. And the model is capable of predicting separate docking scores of separate ligands, maybe future ligands that are not yet maybe performed in a particular experiment, maybe ligands that you have not seen before, and maybe you have, I don't know, maybe 1 million, 10 million of them. You can use the model to calculate the docking scores.
Thank you, Aisha.
Okay, thank you so much.
Okay. Onur, do you have any questions?
Oh, yes. Thank you for the good presentation. I have a very similar question. Actually, perhaps you can discuss very shortly. First, I agree with Dr. Aisha's comment. So what you're doing right now is comparing your workflow, your approach to AutoDocVina results.
Yes.
So why not use experimental binding affinities? So you have mentioned that with AutoDocVina, you are able to generate a lot of binding affinities because you need that many.
Yes.
But I think eventually you will need to compare your approach to experimental data, not AutoDocVina, because AutoDocVina, yes, it predicts something, but its prediction cannot be taken as the best, as the actual binding affinity. That has always, that needs to be validated. Well, there are databases that includes experimental binding affinity values, for example, binding database or KMBL. And there are also derivative databases that includes protein structures as well as ligands, for example, PDB bind CN. So I think if you could incorporate those databases as well in your workflow, that would be very helpful. So yeah, that was it. Actually, it was not a question. It was just a comment.
Yeah, I see. Thank you very much.
Okay, thank you, Onur and Abdus Salam. This is not in your defense, but just wanted to share something. Sometimes these kinds of machine learning dockings are being performed to forecast what the scorings of ligands will be without doing the dockings. That's why you want to do it, yeah? So to search basically for subsets of ligands within these millions of molecules containing compound libraries, to select the ones to dock because you already want to try to foresee what the dockings will be. So in that case, it would be rational to compare it with the Vina scorings. But Onur is also, of course, very right. The real validation would be the active list database and to perform arrangement scorings, of course. Thank you very much, Abdul Salam. So we have two more poster presentations and one oral presentation. I continue to the poster presentation by Hamza Umut. So if we can have that poster online. Yes, that's the one. So let's see it.
Presentation. Thank you for your time. I'm Hamza Umut Karakurt from Gebze Technical University and Media Technology Solutions. Today I will present a computational analysis of transcriptome alterations in the response of SARS-CoV-2 infection in the context of protein-protein interaction networks. And our analysis identified the importance of matrix metalloproteinases in the response of SARS-CoV-2 infection. As we all know, SARS-CoV-2 is a novel coronavirus that causes an acute respiratory disease and it emerged in China and spread globally. It is a really highly contagious virus and it has a long incubation time. Because of that, it requires dramatic lockdown measures. Structural analysis indicated that the receptor for that virus to bind is angiotensin converting enzyme 2 receptor, ACA2, in humans, and the lungs, and especially lung epithelial cells, are the main targets for this virus. Viral infections causes cytokines to emerge as immune response, but as known, cytokine storm is one of the main causes of mortality in patients. Cytokine storm, or hypercytokinemia, is a phenomenon in humans and other animals in which the innate immune system causes an uncontrolled and excessive release of cytokines. Naturally, cytokines are part of the immune system, but their sudden release in high quantities can cause multiple organ failure. In this study, we used the publicly available RNA-seq data from cell lines of mock-infected and SARS-CoV-2 infected bronchial epithelial cells, and we aligned the FASTQ files to human transcriptome, and we applied differential expression analysis, enrichment analysis, and integrated data to a protein-protein interaction network via price-collecting Stalin forest algorithm, and identified communities and clusters. And we worked on the clusters and communities that overlap. First, as you can see in our Volcano plot, we identified 217 genes as significantly changed. These significantly changed genes are highly associated with the signaling pathways, which are highly associated with viral infections. You can see here CADD pathways. And transcriptome data integrated to protein-protein interaction network from string database via PCSF algorithm. We used fold changes of significantly altered genes to construct the infection-specific protein-protein interaction network. Here we use low-vein community detection algorithm to identify the communities, and we applied enrichment analysis to each community here to identify which community should be focused. We tried to find the community with the highest association with the viral infection and immune response. So you can see protein communities 1, 3, and 5 are enriched in signaling pathways and biological processes known to respond in virus. After that, we used cluster 1 algorithm, network clustering algorithm, to detect the significantly functional motifs in the protein-protein interaction network. Actually, these are two different approaches. As you can see, all of the nodes belong to a cluster in the community detection algorithm, but here we identified functional modules. As you can see, this 3 cluster comes from single community. Here it's the biggest community in the low-vein community detection results, but these 2 actually are our main focus. This cluster and the associated proteins are studied before, and they're associated with the SARS-CoV-2 response in the literature. But when we completed that study, there wasn't any literature proof that shows the metalloproteinases may act in the response of SARS-CoV-2 virus. You can see matrix metalloproteinase 9 and 1. It's connected to 2 chemokines, actually, the part of cytokine families. And we did a literature survey about that. And previously, it was reported that deficiency of matrix metalloproteinase 9 in mice is reduced the severity in the influenza virus, in the IAV infection in mice. And taken together with the previous studies about this matrix metalloproteinase in the virus response, we create the hypothesis that the matrix metalloproteinases may act as an important factors in the response to SARS-CoV-2 virus. So in the October 2021, a research group from Sao Paulo, Brazil, conducted a research, and they compared 53 COVID-19 patients and 27 controls. And they saw that the MMP2 and MMP9 levels in the plasma of the patients are altered, significantly altered, and also MMP2 and MMP9 levels are associated, are highly correlated with the mortality and the in-hospital death. So taken together, in a manner, our in silico hypothesis proven experimentally here. And for more details, you can check our published paper in Turkish Journal of Biology, COVID-19 special issue. And here are my contact details. You can write anything you want, anytime you want. And here are my references, and thank you for your time. If you have any questions, I will gladly try to answer. Thank you.
Thank you very much for the nice presentation. Let's continue to the questions. Let's start with Onur this time. Onur, if you're available.
Yes, but I don't have any questions for you.
Okay, thank you very much.
It's nice.
Aisha.
It was a nice presentation. Thank you. I would definitely like to ask, what do you think is the impact of your research?
As an impact, as I said, matrix metalloproteinases as a family, actually their connection to cytokines and the response to virus has been shown before. But actually, by impact, as I said, that this kind of applications using integrating the transcriptome data, information from RNA, and integrating it to protein level data, to a protein-protein interaction network, and by using clustering analysis, actually we can clearly say we can create in silico hypothesis that can be proven right in the response of SARS-CoV-2. Actually, it was the first RNA-seq data that published in the NCBI gene expression omnibus. As I said, it was a cell culture data. But actually, if you read our publication, you can see that actually we offered some possible drug targets on the matrix metalloproteinases, because as you know, there's some aches on the bones, and the bone connection is one of the symptoms of the SARS-CoV-2 virus, especially in the COVID-19 patients. And glycans were one of the possible drugs that can be used to decrease the symptoms. And as I know from the literature, actually for other diseases, not viral diseases, they are used to decrease the pain in the bones for some patients. You know, arthritis, rheumatoid arthritis patients. So actually, we also used some metabolic modeling approaches called reporter metabolite analysis and reporter pathway analysis. And taken together, we also say that we also saw that glucose aminoglycans were regulated in the response. Actually, glucose aminoglycan synthesis and keratin sulfate synthesis were significantly altered in the response. And also these metabolites are also regulating the matrix metalloproteinases. So actually our hypothesis was in the beginning, if we use drugs, possible drug molecules to regulate the matrix metalloproteinases, maybe we can decrease the possibility of cytokine storm. Okay, so our hypothesis, and in a manner of experimental approaches, the paper I mentioned, we show us if the altered levels of matrix metalloproteinases 9 and 2 is correlated with in-hospital deaths and mortality.
Okay, thank you very much, Hamza, for this presentation and the Q&A session. Aisha, sorry, did you have more questions? Aisha? I don't think so. Okay.
I'm here, nothing.
Okay, thank you very much, Aisha. Okay, so let's continue with the next poster presentation. It will be the last poster presentation today. Afterwards, one oral presentation remains. It's the poster presentation by Damanba. So can we have it on screen? This the one? Well, the floor is yours.
Hello, I am Daman Khalindo, and at this 5th International Symposium on Bioinformatics 2021, I'll be talking about the internal transcribed spacer 2 secondary structure analysis of fasciola species from semi-wild ruminants of Northeast India. And Northeast India and is bounded by countries like China, Nepal, Bhutan, Bangladesh, and Myanmar. Now, when you think about India, it is represented mostly by these faces. However, in Northeast India, in the Eastern Himalayan region, the people look somewhat very different. And why this is relevant, these pictures are relevant, because my study, or this study that I'm going to present, is related to the culture, lifestyle, and food habits of the people that reside in this part of the country. So what's fascioliasis? Fascioliasis is a liver rot disease that is caused by a parasite known as fasciola, and it has two species, namely Fasciola hepatica, and the other one is Fasciola gigantica. Now, when you take a look with hepatica and gigantica, you see that the differences are there, especially with morphology. The hepatica is broad, small and stout, whereas gigantica they are much longer and narrower. So materials and methods, there will be field data collection from wide altitudinal range. Then well lab is there, as well as the dry lab methods. And then when we take a look at the collection sites, these are the two semi-wild ruminants that abound in Northeast India. One is the method known as Bos frontalis, and the one is also known as Bos gruniens. Before we begin with the results of the bi-Asian tree, what is to be noted is that the DNA generated from the parasites are sequenced for ITS2. The ITS2 are then put through the ITS2 databases, and only the complete sequences are taken. So out of the 6 isolates that we got, we found out that 4 of the isolates are in the clade representing Fasciola hepatica, whereas the remaining 2 are seen in the clade of the Fasciola gigantica. Now, what is interesting here is that also there is an African and Fasciola gigantica clade, as well as the Asian Fasciola gigantica clade, which is seen in this phylogeny of the ITS2 through Mr. Bias. Now, looking at the secondary structure or the morphometrics of the ITS2 of this parasite, we see that there are 4 helices with the helix 2 showing UU mismatch, and then the presence of motifs in the helix 3. Now, looking at the helix 3, the different motifs that we see in the helix 3, we see that especially with UGG motif, in 2 of our isolates, they are present only 8 in number, which corresponds to Fasciola gigantica, whereas the remaining 4, they have 9 UGG motifs, which corresponds to Fasciola hepatica. So clearly we have Fasciola hepatica and Fasciola gigantica in our midst in Northeast India. This was done using Omega 7. Now, looking at the results, of the secondary structure tree, which was created first and foremost generated by RNA fold, and then this was created by ProfDist, and then we see that we use the dot and bracket sequences that we got from RNA fold. We see that the centroid structures show better resolving power as compared to the minimum folding energy structures. Because why? Because with the centroid structures, we see that the African gigantica clade and the Asian gigantica clade is better resolved as compared to the minimum folding energy structures. Now, this next slide shows a comparative consensus secondary structure generated from fossil of our isolates and the standard Fasciola hepatica, as well as with the standard African and Asian Fasciola gigantica. Now, in the first picture, we see that there are no differences between our isolates, Fasciola hepatica isolates, with the Fasciola hepatica, standard Fasciola hepatica. However, there are 4 green boxes that you see. Now, those green boxes represent the differences between our Fasciola gigantica isolates with the standard Fasciola hepatica. Now, in the image B, we see that there are 2 differences between our Fasciola gigantica with the African Fasciola gigantica, and also the 4 orange boxes represents our Fasciola hepatica differences with the African Fasciola gigantica. When we take a look at the next picture that you see, there are no differences between our Fasciola gigantica isolates with the Asian Fasciola gigantica, with the standard Fasciola gigantica from Asia. However, there are differences between our isolates, Fasciola hepatica isolates, with the Asian Fasciola gigantica, which is clearly shown in this table in the next slide. So with this, I would like to conclude by saying that the second structure or the molecular morphometrics of ITS2 is very useful, because the secondary structure tree corroborates the Bayesian tree, and then it also shows the different helical structures, and the helix tree proved to be very useful for species discrimination, and the centroid structure is much better than the minimum folding energy as far as resolving the phylogeny of this parasite that we are using. With this, I would say thank you.
Okay, thank you very much. Unfortunately, it was very fast and very difficult to understand. So Aisha, I hope you have any questions. Aisha, are you there?
Yeah, I'm there. He's very fast, but I couldn't understand the correlation between the phylogenetic analysis and the secondary structure prediction. How are you relating it? Because you just jumped from somewhere. And your explanation was also, I couldn't follow, you were too fast. Wait, are you there?
Yeah, I'm here. Can you hear me?
Take the time. Yeah, I can hear you.
Okay. Yeah. So what happened with the phylogenetic analysis is this, that with the ITS2, we have two species of Fasciola in over there, that is Fasciola hepatica and Fasciola gigantica. So when we take a look at the secondary structure, there is the centroid structure that we have taken as well as the minimum folding energy. So the minimum folding energy does not corroborate or doesn't show the identical structure with the phylogeny that we have, but the centroid structure shows that, which corroborates the same phylogeny as we get from Mr. Bias. Now, interesting thing is that in India itself, there is no Fasciola hepatica. So this is the first of its kind, because this is also Northeast India's high elevation areas, closer to Himalayas. So these samples are very different samples, and they are also very rare samples, so to say. So they give us an insight of what is happening in this part of the country when it comes with this parasite. Because in other parts in China and other parts of the bordering areas, Fasciola hepatica is there, but in the rest of India, it's not reported. So this is the first time this has been reported.
Okay.
Yeah.
Okay. Dr. Onar has a question.
Thank you for the presentation, but I thought it was really fast and it was a bit difficult to follow. But yeah, I had the same question as Dr. Ayesha, so thank you for clarifying that.
Thank you.
Thank you.
Okay. Let's continue then to the last presentation, which will be an oral presentation by Mahendra, and it's on screen, so let's play it.
Good morning, all of you. Myself, Afzal Ansari, Research Associate from ICMR NERT, Jabalpur. And my current topic is, presentation is genomic characterization and phylogenetic analysis of SARS-CoV-2 strains in Madhya Pradesh. So, first of all, SARS-CoV-2, we all know SARS-CoV-2 genomes. First COVID-19 case denoted in Wuhan city in China, late 2019, December 2019, and it spread all over the world in just a few weeks, but WHO declared it pandemic in March 2020. So, and there are lots of different types of category, like variant of concern, variant of interest, and variant of monitoring. So out of three types of variants, the variant of concern is more virulence and more dangerous for health, or more virulence and more spreadable for in this lineage, which is called the variant of concern. And there are 5 types of variant of concern here. 5 types. One is alpha, beta, gamma here, alpha, beta, gamma, delta, and currently Omicron is coming, which is 26 November 2021. And so our aim is to perform the whole genome sequencing of 8 earliest reported cases of COVID-19 in Madhya Pradesh. So to perform the comparative genomics analysis to this initial case with all the other available genomes from Madhya Pradesh state, determine the genetic diversities. So first of all, we collect the samples. We collect the sample nasopharyngeal and oropharyngeal and RNA extraction and test for the qPCRs screening. First, we screen the samples, patients who is the suspected or not. We screen that patients and isolate them. If they're positive, indicate in the red. If they're positive, we isolate them and we collect some earlier samples, which is March 2020 or April 2020, suspected or being positive of SARS-CoV-2. So after that, we sequence them samples using Illumina, and after sequencing, we analyze with using bioinformatics tools. Next, also we download sequence from submitted to the Wuhan GSAID, which is belong to the Madhya Pradesh genomes. So we compare with them, and after comparing, we find out the phylogenetic analysis. Compare using phylogenetic analysis. Next trend. So here the result we shown here our 18 samples in next slide. So here one samples like here the ICMR NERT is 14 samples. Only one sample is not present D614G mutations. So these are the mutations, yellow, green, and different lines. But gray line indicate the missing regions, which is not covered by the sequence, by the reference sequence, by the sequenced samples. After that, we here the present all 18 samples and their lineage and their clades. All are 20A clade, next string clade, but only one sample, which is S14, which is 19A clade, and all samples belongs to the common countries like UAE, Saudi Arabia, and USA, UK and USA. So after phylogenetic analysis, our 18 samples in A part, we see there our 18 samples are phylogenetic. 8 are clustered together, which is a B.1.95, and 6 are common B.1.36, B.1, and B.1.36 2 samples, and B is 1. And B.6 is 1 sample, which is S14, which is closest to Wuhan. And now in these sections, we see here our 18 sample denoted is here, color with labels with yellow. Now here we see here the most outer layer is indicate the Pango lineage, and here we show the red line, which is indicate the delta, orange indicate the kappa, and this blue lines indicate the alpha. So also we seen here the B.1.95 is cluster differently in all over the 861 samples. So finally, we found after the phylogenetic analysis, there the 5 lineages indicated in our samples, and one lineage B.1.195 is associated with important cases has not been observed outside the Jabalpur, and that samples are indicate with travel history, international travel history. So it's uniquely different. And we found 44 different SARS-CoV-2 pangolin lineages, 44 different types of SARS-CoV-2 pangolin lineages. Out of 44, we found here the alpha, which is the most highest, B.22%, and beta very less, only 2 samples out of 861, 0.23%, and delta we found around 12%. And here the others samples are, we can see. Conclusion is that this studies, phylogenetic analysis is strain in this study point out the multiple introduction of SARS-CoV-2 virus in Madhya Pradesh, and conclude the genomic surveillance strategy are needed to improve the monitoring and understanding the current SARS-CoV-2 pandemics. Because of sequencing is the most important. Sequencing is most important to find out which current lineage is found or not. If we sequence the out of the positive samples, we sequence almost 5%, then we found which lineage is currently virulence or not. If we found that lineage is virulence, then we highlight them, and also we isolate them and lockdown and others precaution we can measures. I also acknowledge to ICMR, GSAID, WHO, and DBT, and this is our institutes, ICMR, which is present in Jabalpur, Madhya Pradesh, in India, and our lab members are present here. There is our PI Pushpinder Singh, which is the middle one. Thank you.
Thank you very much for the presentation. So let's go to the question section. Onur, can I have your questions if you have any?
Yes, I have one question. Thank you for this presentation. On your results slide, you showed some amino acid and nucleotide substitution. Was it correct? Yeah, so there was some nucleotide substitutions on one of the slides. Yeah, so which proteins have you, was your analysis based on only spike protein, SARS-CoV-2? I could not get, if you could increase the sound, the voice of your microphone.
Hello. Hello.
Yes, I can hear. Yes.
I hear Reptile, and here you can see that this is the total number of genome size of SARS-CoV-2. And also, we cannot hear you. Can you hear it?
I can hear you very clearly, but I cannot hear the presenter. I think there's an issue with his microphone.
Yeah, okay.
So you can perhaps write your answer in the chat.
Hello. Am I audible? Hello.
It's better now.
Afsal, can you
Answer?
increase the microphone, not volume, but sensitivity?
But mutation are in all these, I think, because when we have the complete genome size, and you can see here the green line or this yellow line is present in different regions, like in ORF1A, 1B, and here is spike protein here. So we can see that mutation is all over the genome, and we can see here in table, like nucleotide mutation is 241, which is the upstream side, and here the amino acid mutation, and we denoted with the gene N, ORF1B, ORF3A, and S spike protein. So I think mutations are present in different genes also. Hello.
Okay, I think this was enough. I got my answer. Thank you.
Okay, thank you.
Thank you.
Thank you, Onur. Thank you, Afsal. Aisha.
No, I don't have a question.
Okay, thank you, Aisha.
Thank you for the
thank you, Afsal, for your presentation. So this was, according to my list, the last oral presentation, actually the last presentation of this parallel session. I think we resume at 4:10 with the keynote lecture of Anton Veenstra, but let me give the floor to Birgit. Thank you very much.
Thank you, Professor Dr. Attila, and our honorable judges. I would like to thank all others who put efforts on oral and poster presentations, and all our participants for being here with us. CADD and SARS-CoV-2 presentations are over now. Dear judges, please fill the forms for these oral and poster presentations. Last big data analysis and biotools and general bioinformatics presentation is going on now. If you want, you can change your breakout room to join there. Later on, the next session will begin at 4:10 p.m. according to Turkey's standard time for our last keynote talk by Dr. Anton Veenstra at the main room. See you there.